This is a painting from the 16th century from Lucas Cranach the Elder. It shows the famous fountain of youth. If you drink its water or you bathe in it, you will get health and youth. Every culture, every civilization has dreamed of finding eternal youth. And there's people like Alexander the Great or Ponce de Leon, the explorer, who spent much of their life chasing for the fountain of youth. They didn't find it. But what if there was something to it? What if there was something to this fountain of youth? I will share an absolutely amazing development in aging research that could revolutionize the way we think about aging, and we may treat age-related diseases in the future. It started with experiments that showed a recent number of studies, but growing, that animals, old mice, that share a blood supply with young mice can get rejuvenated. This is similar to what you might see in humans, in Siamese twins. And I know this sounds a bit creepy. But what Tom Rando, a stem cell researcher, in 2007 reported was that old muscle from a mouse can be rejuvenated if it's exposed to young blood through a common circulation. This was reproduced by Amy Wagers at Harvard a few years later, and others then showed that similar rejuvenating effects could be observed in the pancreas, the liver, and the heart. But what I'm most excited about, and several other labs, is that this may even apply to the brain. So what we found is that an old mouse exposed to a young environment in this model called parabiosis shows a younger brain and a brain that functions better. And I repeat, an old mouse that gets young blood through the shared circulation looks younger and functions younger in its brain. So when we get older, we can look at different aspects of human cognition, and you can see on this slide here, we can look at reasoning, verbal ability, and so forth. And up to around age 50 or 60, these functions are all intact. And as I look at the young audience here in the room, we're all still fine. But it's scary to see how all these curves go south. And as we get older, diseases such as Alzheimer's and others may develop. We know that with age, the connections between neurons, the way neurons talk to each other, the synapses, they start to deteriorate, neurons die, the brain starts to shrink, and there's an increased susceptibility for these neurodegenerative diseases. One big problem we have to try to understand how this really works at a very molecular mechanistic level is that we can't study the brains in detail in living people. We can do cognitive tests, we can do imaging, all kinds of sophisticated testing, but we usually have to wait until a person dies to get the brain and look how it really changed through age or in a disease. This is what neuropathologists do, for example. So how about we think the brain of being part of the larger organism? Could we potentially understand more about what happens in the brain at the molecular level if we see the brain as part of the entire body? So if the body ages or gets sick, does that affect the brain? And vice versa, as the brain gets older, does that influence the rest of the body? And what connects all the different tissues in the body is blood. Blood is the tissue that not only carries cells that transport oxygen, for example, the red blood cells, or fight infectious diseases, but it also carries messenger molecules, hormone-like factors that transport information from one cell to another, from one tissue to another, including the brain. So if we look at how the blood changes in disease or age, can we learn something about the brain? We know that as we get older, the blood changes as well. So these hormone-like factors change as we get older. And by and large, factors that we know are required for development of tissues, for maintenance of tissues, they start to decrease as we get older. While factors involved in repair, in injury, and in inflammation, they increase as we get older. So there's this unbalance of good and bad factors, if you will. And to illustrate what we can do potentially with that, I want to talk to you through an experiment that we did. We had almost 300 blood samples from healthy human beings, aged 20 to 89 years of age, and we measured over 100 of these communication factors, these hormone-like proteins that transport information between tissues. And what we noticed first is that between the youngest and the oldest group, there's about half the factors change significantly. So our body lives in a very different environment as we get older when it comes to these factors. And using statistical or bioinformatics programs, we could try to discover those factors that best predict age. In a way, back calculate the relative age of a person. And the way this looks is shown in this graph. So on the one axis, you see the actual age a person lived, the chronological age. So how many years did they live? And then we take these top factors that I showed you, and we calculate what is their relative age, what is their biological age. And what you see is that there is a pretty good correlation. So we can pretty well predict the relative age of a person. But what's really exciting are the outliers, as so often in life. You can see here, the person I highlighted with the green dot is about 70 years of age, but seems to have a biological age, if that's really true, what we're doing here, of only about 45. So is this a person that looks actually much younger their age? But more importantly, is this a person who is maybe at a reduced risk to develop an age-related disease and will have a long life, will live to 100 or more? On the other hand, the person here, highlighted with a red dot, is not even 40, but has a biological age of 65. Is this a person at increased risk of developing an age-related disease? So we're trying in our lab to understand these factors better, and many other groups are trying to understand what are the true aging factors, and can we learn something about them to possibly predict age-related diseases. So what I've shown you so far is simply correlational, right? You can just say, well, these factors change with age, but you don't really know if they do something about aging. So what I'm going to show you now is very remarkable, and it suggests that these factors can actually modulate the age of a tissue. And that's where we come back to this model called parabiosis. So parabiosis is done in mice by surgically connecting the two mice together, and that leads then to a shared blood system where we can now ask, how does the old brain get influenced by exposure to the young blood? And for this purpose, we use young mice that are an equivalency of 20-year-old people and old mice that are roughly 65 years old in human years. What we found is quite remarkable. We find there's more neural stem cells that make new neurons in these old brains. There's an increased activity of the synapses, the connections between neurons. There's more genes expressed that are known to be involved in the formation of new memories. And there is less of this bad inflammation. But we observed that there's no cells entering the brains of these animals. So when we connect them, there's actually no cells going into the old brain in this model. Instead, we reasoned then that it must be the soluble factors. So we could collect simply the soluble fraction of blood, which is called plasma, and injected either young plasma or old plasma in these mice, and we could reproduce these rejuvenating effects. But what we could also do now is we could do memory tests with mice. As mice get older, like us humans, they have memory problems. It's just harder to detect them, but I'll show you in a minute how we do that. But we wanted to take this one step further, one step closer to potentially being relevant to humans. What I'm showing you now is unpublished studies where we used human plasma, young human plasma, and as a control saline, and injected it into old mice and asked, can we again rejuvenate these old mice? Can we make them smarter? And to do this, we use a test. It's called a Barnes maze. This is a big table that has lots of holes in it, and there's guide marks around it. And there's a bright light, as on this stage here. The mice hate this, and they try to escape and find the single hole that you see pointed at with an arrow, where a tube is mounted underneath where they can escape and feel comfortable in a dark hole. So we teach them over several days to find this based on these cues in the space. And you can compare this for humans to find your car in a parking lot after a busy day of shopping. Many of us have probably some problems sometime with that. So let's look at an old mouse here. This is an old mouse that has memory problems, as you notice in a moment. It just looks into every hole, but it didn't form this spatial map that would remind itself where it was the previous trial or the last day. In stark contrast, this mouse here is a sibling. It has the same age, but it was treated with young human plasma for 3 weeks with small injections every 3 days. And as you noticed, it almost looks around, where am I? And then walks straight to that hole and escapes. So it could remember where that hole was. So by all means, this old mouse seems to be rejuvenated. It functions more like a younger mouse. And it also suggests that there is something not only in young mouse plasma, but in young human plasma that has the capacity to help this old brain. So to summarize, we find the old mouse and its brain in particular are malleable. They're not set in stone. We can actually change them. They can be rejuvenated. Young blood factors can reverse aging, and what I didn't show you, in this model, the young mouse actually suffers from exposure to the old. So there's old blood factors that can accelerate aging. And most importantly, humans may have similar factors, because we can take young human blood and have the similar effect. Old human blood, I didn't show you, does not have this effect. It does not make the mice younger. So is this magic transferable to humans? We're running a small clinical study at Stanford where we treat Alzheimer's patients with mild disease with a pint of young plasma from young volunteers, 20-year-olds, and do this once a week for 4 weeks. And then we look at their brains with imaging, we test them cognitively, and we ask their caregivers for daily activities of living. What we hope is that there are some signs of improvement by this treatment. And if that's the case, that could give us hope that what I showed you works in mice might also work in humans. Now, I don't think we will live forever, but maybe we discovered that the fountain of youth is actually within us, and it has just dried out. And if we can turn it back on a little bit, maybe we can find the factors that are mediating these effects. We can produce these factors synthetically, and we can treat diseases of aging, such as Alzheimer's disease or other dementias. Thank you very much.